using the sediments and diatoms. So Madagascar was located in the southeastern of Africa and constitute a biogeographical entity different from all the others of our planet Earth. So it has a various landscape and has a multiple geographical zone, but I'm interested in the Central Highlands because this region is now largely deforested and devoid of native vegetation and dominated by grasslands and the steppe. So I chose my studied area because as we see here, almost of the subfossil sites of Madagascar are located in the southwestern part and in the west, but there are some localities in the Central Highlands, and Tsaramody site was found during our expedition with a Cenozoic project, was led by Dr. Sammons, and this site is a result of the volcanic activity during the Upper Miocene to the Holocene. So we see here this site actually the rest of the vegetation, and this site is the population used like rice field and the cabbage field. And why I use diatoms? Because diatoms are found in all aquatic environment, and it's an excellent bioindicator for paleoenvironment reconstruction. And I combine the diatom analysis with sedimentological analysis to reconstruct the past climatic conditions and the hydrological characteristic of the environment through times. Because we know each sedimentary layer contains chemical biological indicators and which make it possible to reconstruct the past environment. So as the methods, we did the sampling in this site. The sediments were collected from the outcrop excavated in this area, and then we did the coring because the sediments are soft, and all samples then shipped to the laboratory of the micropaleontology and paleobotany at the University of Antananarivo for chemical treatment in order to separate the sediment and the atoms. So in the lab for chemical treatment, we use the standard protocol for the atom treatment. And then we did an observation and we count each taxa because each taxa has an ecological significance for the habitat considered, and it's necessary to have the atom assemblages after identification. So I use this the atom assemblages established by Gassi and al. And these assemblages was used to know the datum associations and their distributions. So as a result, sedimentological results from this site are composed of the clay and travertine peat, clay peat, and vegetated soil. So as we see on the outcrop, this layer are almost the dark layer, and these sediments are rich in organic matter, and it indicates the humid climate with high biological productivity. And the radiocarbon dating indicates that these sediments was deposited during the Pleistocene to Holocene. And in Tsaramody, 12 species of diatoms were encountered, and these are the samples of an example of this diatom species. And the assemblages of this diatom group belongs to group 1 and 2. So we see the change in diatom assemblages. It reflects in the sequence of environmental transformation. It begins with a hypersaline water to the high altitude lake and pit bogs to a marshy environment, and then this tree environment characteristic is repeat again through the highest level. So the absence of the diatoms in the level 1 means that the crystal diatoms dissolve rapidly before they can be incorporated into the sediments. So this transition indicates significant change in water chemistry, as well as the fluctuation in the lake level, the temperature variation, likely driven by a regional climate variability, including shifting between the wetter to drier periods. So we see here the environmental reconstruction of surrounding this site during the quaternary periods. So the dominance of the panel diatoms suggests a shallow lacustrine environment and with high organic matter and high biological productivity, and this data indicates that the site would have been a permanently wet area, and these knowledges could provide details into past
I guess we lost Miura.
We have a technical issues, Miura.
Miura?
Let's wait some minutes if Miura can recover the internet.
If not, we could continue, maybe.
Yes, maybe someone has some questions, and I can write them in the chat for Miura.